Thank you. I'm excited to be able to share this work with everyone. So familial hypercholesterolemia is a disorder of LDL metabolism. So LDLs are particles that carry cholesterol through our bloodstream. And what happens is if we have a genetic predisposition based on a defect in one of these three genes, LDLR, ApoB, or PCSK9, our bodies aren't able to bring this LDL into the cells and get it out of our circulation. And so because of these elevated LDL levels, FH patients are predisposed to atherosclerosis. So what happens here is that LDL can enter the artery wall. There it can cause plaque buildup. So this is sort of an inflammatory reaction that results. Plaques can grow. This restricts blood flow in your arteries, and this can lead to stroke, heart attack, and often death. So what's really important in FH is to start on therapeutic cholesterol-lowering therapies early on so that you can prevent or at least delay the onset of atherosclerosis. So to be able to do that, we need, obviously, good genetic diagnostics to be able to identify these patients early on. And for that, we need to be able to understand genetics. And the reason for that is because cholesterol alone does not serve as an appropriate marker for FH. And C. Catharesin's group showed this nicely, that patients who have an LDLR mutation or an FH mutation are predisposed to coronary artery disease sort of regardless of their LDL level. So you can see this on the left if we look at the odds ratio for coronary artery disease across the distribution of LDL levels for wild type people in blue or FH carriers in purple. And the takeaway here is really threefold. So the first is that not everybody with an FH mutation actually presents with a really severe clinically elevated LDL level that we can identify with just cholesterol measurements. The second is that despite having what would look like normal-ish levels, they're at an increased risk of disease. And the third is that the therapeutic guidelines for people with FH mutations are actually a bit different. So the LDL levels that your clinician wants you walking around with are about 25% lower if you have an FH mutation compared to the rest of the population. So we obviously wanted to be able to do clinical genetics for FH and to improve this. And one of the major things that we're missing here is, of course, new sources of evidence to classify variants of uncertain significance. I don't think I need to really convince anyone here of that. So this led us to doing a sequence function study of LDLR, and I'll tell you a bit about how we did that at scale. And an interesting story that's come from that. So just a bit about what the LDLR actually does. So this is a receptor that presents on the cell surface. And so here it will bind to the ApoB apolipoprotein in the LDL particle. So this is the protein constituent of the LDL particle. This complex will get internalized. LDLR can be recycled to the cell surface. Once it's released LDL, LDL can be degraded. And so this is sort of how LDL gets out of the bloodstream. And now this is the biology that we're really targeting with our assay. So in our assay, we're actually measuring how variants affect LDL uptake. So how do we do this? We have a barcoded library of LDLR coding variants, which we integrate into an LDLR knockout HeLa cell line, which has a genomically integrated BXB1 landing pad developed by Kenny Matreyek and Doug Fowler's lab, or I guess used to be in Doug Fowler's lab. And so this allows us to ensure that only one variant is getting into each of our cells. And then because we're using a fluorescent LDL molecule, we can separate variants based on fluorescence-activated cell sorting. And then from each of those bins, we can demultiplex our libraries by sequencing barcodes, and this leads us to a functional interpretation. So we've now done this for the entirety of the LDL receptor. I just want to take a pause here and acknowledge Atina Cote, who's not here today, but who played a huge part in this work. So just a quick overview. We've seen these enough, but x-axis here is amino acid position. Y-axis is amino acid substitution. And so that means that each of these little squares here is one given variant for which we have a functional score. And these functional scores are colored based on white denoting a functional score that is akin to a synonymous function, and blue is damaging. So if you look at this long enough, you can get some pretty cool biology. One of our favorite parts of the protein is the ligand binding domain. So in the crystal structure on the left, this is that gray arm that's wrapping around the protein. And so this domain is made up of 7 highly conserved and highly similar LA repeats, which are very important for binding LDL and releasing LDL. So when you disrupt these, you end up with impaired LDL uptake. So we can get at some of the biology that's happening in some of these with quite a lot of sensitivity. So this is a calcium coordination cage in LA5. Each of these LA repeats has 1 calcium ion that's important for the structure of the repeat and also the binding and release of LDL particles. So there are 6 residues that are coordinating this cage. 4 of these contribute a side chain carboxyl group. These obviously can't be substituted, so these do not tolerate substitution. Those are the 3 aspartic acids and the 1 glutamic acid. Interestingly, the glycine, which is shown in the blow up there, contributes a main chain. We find that this tolerates virtually all substitution. And then the tryptophan on the bottom left, which is also contributing a main chain carboxyl group, we think the reason that this can't be substituted is because it's actually involved in a pi stacking interaction with another domain of the protein. So all that to say, we can measure variance in this certain parts of the genes with quite a lot of sensitivity. Of course, for other parts of the ligand binding domain, we're kind of out to lunch. So in our assay, LA repeats 1, 2, and 6 tolerate variation, and that includes variation that we know is pathogenic. We've seen it in the literature. We've seen it in population cohorts. So this is kind of a major bummer when this is your finding. And so it got us thinking about, okay, what could be happening in our assay that we're actually seeing this? So far I've really only talked about LDL. This is, of course, not the only lipoprotein in circulation. There are many of these at different stages, different sizes, different constitutions. And the one of relevance here is VLDL. So this is a larger lipoprotein, which has more apolipoproteins in it, and it's about 50% triglycerides. So it's got more triglycerides in it than LDL. And LDLR is able to interact with this lipoprotein. And so what we thought might be happening here is that those LA repeats, which we're not picking up in our assay, might actually be involved in VLDL binding and not LDL binding. So what we did is a second map in the presence of VLDL. So here what we're measuring is LDL uptake, but there's VLDL floating around in the media. And what we found from this is that our hypothesis was actually completely wrong. So where we thought these LA repeats might be binding to VLDL, what they're actually seem to be important for is conferring a specificity to LDL. So on the top is our map where we just have LDL, there's no VLDL in culture. On the bottom, when we add VLDL, we start to see these stripes show up, and these are at positions that we know to be important for LDL binding. So we think that this might have a sensitizing effect in culture. So I don't really have a mechanism for this. I wish I did. It's hard to model these things because they're large and complex, but we're working on it. And at minimum, this is an interesting finding that's come from doing maps in different physiologically relevant environments. Okay, so I'm going to completely switch gear and talk about some of the work we've been doing using our functional scores to enhance FH risk prediction in the UK Biobank. So first off, our map does a pretty nice job of separating ClinVar pathogenic from benign variants, which is great because we want to be able to use this to help reclassifying VUSs. But we also wanted to ask, could our scores be useful for variants that already had classifications? So ClinVar LP, P, or pathogenic variants for which we already have a classification. And so we thought, okay, maybe we can integrate scores with these variants and start looking at FH risk prediction. So how do we predict FH in a population-based cohort? Well, there are these really nice points-based criteria that have been laid out by the Dutch Lipid Clinic that look at a combination of family history, clinical presentation, LDL cholesterol levels, and FH genotype. And so we've adapted these in the UK Biobank to sort of get a sense of how likely do we think someone has, what would be a diagnosis of FH in the clinic. And so then we looked at using these evidence types to select the qualifying variants and then look at the odds ratio of disease for participants with those variants. And so you can see that if we're just using a damaging LDL uptake score, just using ClinVar pathogenic, likely pathogenic annotations, we get an odds ratio that's pretty similar. But if we then use LDL uptake scores to sort of refine that set and look at the set of pathogenic variants that have these stronger phenotypes, then we get a different profile of risk. And so this sort of suggests that functional evidence can still be interesting even where we have a known pathogenic variant. So of course, if we want to think about risk, we can't really do that properly without also integrating common genetic variation into our background. And so we've done this using the PRSs in the UK Biobank. So what we've done is looked at the odds of having an FH phenotype, so clinically elevated LDL or being on a cholesterol-lowering medication, based on your common and rare variant genetics. All right, so in black, I'm showing rare variant types, so whether you have a damaging LDLR variant, and then gray is wild type, and then this is separated on the left based on PRS percentile, so low percentile is protective, high percentile is damaging, and on the right, this is the exact same thing, just across the spectrum of polygenic risk score. And so what we see is that really these rare large effect monogenic variants are doing a lot of the work in terms of conferring risk, which of course makes sense, but our PRSs are actually acting as a conditioner, right? So if we look especially on the left, comparing people with damaging variants in the bottom and upper PRS percentiles, we get a really different picture of risk. And so it's interesting to be able to combine these together when we think about disease risk prediction. And of course, we can do the same thing thinking about time to CAD event, so time to having a coronary artery disease event. So in black is your reference. Interestingly, a low PRS score does actually confer a protective effect when we're thinking about time to CAD. Now, what's striking here is that actually high PRS and damaging MAP score shown in pink actually have a pretty similar effect on time to CAD. But when we combine these and look at, okay, for participants that have a detrimental monogenic variant as well as a polygenic background, then we start to get a pretty different picture of risk. And so this sort of illustrates how important it is to think about disease risk across the spectrum of allele frequencies. Now, we've done this in the UK Biobank. We're moving to a diverse population-based cohort in all of us. So the great thing about MAP scores, of course, is that they're ancestry agnostic, and so we can do this. And we're working with Dan Roden and Megan Lancaster at Vanderbilt to extend this risk prediction to all of us. And this, of course, means also integrating population-specific PRSs into that risk prediction. Okay, so with that, in summary, we've generated a full-length sequence function map of LDLR. We've shown that changing environments can uncover some interesting biomolecular dependencies, and that integrating common and rare variation does in fact refine risk prediction. And so with that, I'd like to thank, of course, Fritz and Atina and our CardioVar members, Dan Roden and Megan Lancaster. Thank you.
Wonderful. Thank you, Daniel. That was great. Questions from the floor? While people are warming up, can I ask you about benchmarking of your assay for your MAVE? What have people done historically to understand the consequence of receptor mutations? And if you were evaluating your wonderful map against those more traditional methods, how do they map? How do they benchmark? How do they line up?
So I mean, the studies that go looking at LDLR, they go a long way back. So a lot of this is sort of old school lipoprotein biochemistry. Mm-hmm. So if we compare to those, we do pick up a lot of the same information in those LA repeats. So when we look at the biochemistry there, and then of course we've mostly looked at clinical variation to see how we do against that. Yeah. Mm-hmm. Yeah.
I guess I was curious about some of the variants that you were unable to detect with your large-scale approach, and whether they had different mechanisms that had been seen with alternative methods.
So unfortunately, what's sort of, I think, happened there from the literature is that these haven't been uncovered at the depth that we would like. So what sort of happened is that because these LA repeats are all so similar, part of what's happened, I think, is that we assume that, okay, a damaging variant in one, if it's in that conserved aspartic acid, in another LA repeat is the same. And so in fact, if you look at some of the genetic testing guidelines, they say, okay, if you have a variant in one of these positions, in any of the LA repeats, we think that that's damaging, right? So this hasn't been teased out at the depth that we would like. There's been studies that delete entire LA repeats and look at sort of which ones have an impact on function, but that hasn't been super informative for us, unfortunately. Yeah.
Thank you. Over there.
Willie Buxer, I love how you and several people have now showed the same map, but in different contexts, like in your case, plus and minus the VLDR. Yeah. Have you thought of, or have any of us thought of, like, how do we look at the variance, right? So obviously, if you had like 10 replicates in the same exact context, there would be some changes in the heat map coloration, but how can we all like start to look at these multiple maps of the same thing and understand, you know, what's just variance and what's really like a big difference?
Yeah, that's a great question, and also one that we're kind of in the process of figuring out because we do need to compare these properly. I don't have a good answer for you beyond that we need to think about how to scale these properly, really. But I think that that's something that we need to do as a community, is be able to, if we release a map, show that all of our maps are on the same scale so that we can at least compare between them, effectively. Yeah.
Melina, do you have a question?
No, hi, Melina.
Phenomenal talk as usual, Daniel. I don't see. Awesome work. I really love it. So given this context specificity, which is just really beautiful to see, and I think we discussed about this previously, I totally acknowledge the kind of limitations of implementing this in different disease-relevant cellular model systems. Do you, what do you think is the path forward in integrating such MAVE systems in, you know, not even primary human cells, but cell types and cell states that are specific given the context dependency that you're evaluating?
Yeah, absolutely. I would have loved to be able to do this in HepG2 cells, in hepatocyte-derived cell lines. That was initially what we set out to do, but it turns out that there were some technical limitations in terms of doing them at scale. But I think a natural next step to this would, be at least validating these findings in the right cell types. I think they can still be cancer cell types, at least to start. But yeah, that would be a nice next step for us. We would love to get them to scale and to have a different graduate student redo these maps in HepG2s. Yeah.
Wonderful, that's fantastic. Well, I'd like to thank Daniel again and all of the speakers from the session. It's been wonderful to hear so many different projects.